Okay, I'm Garth Nicolson. I'm going to talk about the Fluid-Mosaic membrane model at 50 plus years. So I'm a professor emeritus of pathology and laboratory medicine, and I'm the founder and president of the Institute for Molecular Medicine. I consult with a couple of companies, and you can find my publications at ResearchGate, at least 600 out of my 750. So I'm going to talk a little bit about the Fluid-Mosaic membrane at 50 plus years. It's some principles of it, domain features of biological membranes, control over membrane surfaces, and the role of lipids in membrane dynamics. Obviously, in 15 minutes, I can't cover that, but you can go to my mini review that I wrote on this subject in BBA biomembranes, and that's shown here. And basically, the Fluid-Mosaic membrane came out on a timeline of really rich history of research in biological membranes. But there was a rich history before, which I don't really have time to go into. But I do want to mention that at the time when I worked on that, I actually was a graduate student at the time, the Danieli-Davson model of membranes, or the tri-layer model of membranes, was the textbook acceptable model. And Green and Benson also had a model based on lipoprotein units, which I'll come into. With my thesis advisor, S. Jonathan Singer, we produced a Fluid-Mosaic membrane model, which was a major part of my thesis, and published that in Science, and that's kind of become the textbook model. But if we compare the tri-layer Danieli-Davson model with the Benson-Green lipoprotein model and the Fluid-Mosaic model over here at the right, really only the Fluid-Mosaic model fit with the available data at the time, and this mainly was the late 60s, very early 70s. The other models really fell down in certain respects, and it's kind of shown here. So the textbook model for membranes has ended up to be the Fluid-Mosaic model, and there are obviously a number of forms in various textbooks. The models are varying degrees of complexity, but basically they have the same basic principles. And I'm going to discuss that now. And those principles are that basically biological membranes are held together by non-covalent forces driven by the hydrophobic interactions or hydrophobic effect and van der Waals forces. And that's a major emphasis of the model of membranes. And of course, it depends upon lipids forming the matrix of the model. And this is discussed by a variety of different authors. I really don't have time to go into it. But the lipids arrange, glycerophospholipids in particular, arrange in lipid bilayers, and this provides the basic matrix and continuity of cellular membranes. And in amphipathic integral membrane proteins and glycoproteins are intercalated asymmetrically into the lipid bilayer, and this occurs during their biosynthesis. So we end up with a variety of different forms of membranes, and this is sort of shown here at the textbook model, just showing basically the asymmetry of it, the fact that the lipids and proteins are considered separate, although there's some tightly bound lipids to the proteins. And this is very important, that there are various classes of membrane proteins. There are integral membrane proteins, which are intercalated into the lipid bilayer, and then there are peripheral membrane proteins, which are peripherally attached, some by covalent bonds, others by interactions with other membrane proteins, and so on. And then there are also what I call membrane-associated proteins, which are basically elements within cells like cytoskeletal elements or extracellular matrix outside of cells, which associate with membranes but are not really part of the membranes. So the membrane components attempt to maximize their molecular interactions, and this occurs by the matching, hydrophobic matching of proteins and lipids. And this is in order to approach the lowest free energy state, which of course is never really approached, but there are different glycerophospholipids, for example, which have different distances, and these match up with different proteins, depending upon the fluid nature of their chains or their non-fluid nature. Different types of proteins can fit into this type of array and are basically free to move if the lipid bilayer that they're intercalated into is fluid, or they're static if it's non-fluid. So membranes are generally organized into various domains or regions of different component compositions, lateral mobilities, lifetimes, and functions. And basically, we didn't know much about this at the time of the origin of Fluid-Mosaic model, and I've worked on this myself and other people have as well, but this is kind of an add-on to the Fluid-Mosaic. And there are a number of add-ons to the Fluid-Mosaic, which is why the model is still relevant, just as long as it's brought up to date. But even in 1976, when I wrote a review on it, I was out of graduate school then. I was an assistant professor at the Salk Institute, and we were looking at membranes as components with different arrays of materials, so they could have aggregations or associations to form domains, for example. And there were also peripheral membrane components shown here as extracellular matrix or cytoskeletal elements, which immobilized components in the membrane. And those immobilized components we didn't think much about in the early 70s, but they became more and more important as time went on. So lipids form naturally into different domains depending upon their composition. And you can even see this by atomic force microscopy, which is shown here by Mortison's work and other people as well, that the different lipids can organize into different lipid islands, more or less. And these have been called lipid rafts or different islands or whatever. But basically, they're different structures that have dynamics, and they're present as domains. So these lipids, for example, can change in their makeup, their composition. They can change in their rotational mobility of individual lipids, their lateral mobility of the domains itself, and whether they're static or moving, and whether they can recombine or break up. So the hydrophobic interactions of lipids and proteins result in molecular sorting. And so this means that membranes are naturally sorted out based upon their hydrophobic, mainly hydrophobic interactions. And this results in variability on the membrane surface. And that variability can control a lot of the different aspects of membrane and their function. So what types of models are suitable to explain biologic phenomena? And I put to you that the Fluid-Mosaic model is the suitable model that can explain the dynamics of membranes, their structure, and also a number of other properties that they have. So with time, for example, the states of membranes change quite radically. They can change in terms of the aggregation states of different components in the membranes. And some of this is shown here from a review by Simmons and collaborators in Cold Harbor Symposium. They can also change in terms of their lateral distributions in membranes. And this is just shown in one study that looked at the lateral display of membrane components and how some are organized into kind of bigger islands and others are more dispersed. And of course, this can change, and the compositions of these can change over time. So the lateral mobilities of membrane proteins can be controlled by various associated complexes, and primarily inside or outside the cell, cytoskeletal elements outside or inside the cell, and extracellular matrix and stromal components outside the cell. And many membranes have this important role of the membranes, a certain static nature of the membranes. They're not all membranes are totally fluid. Matter of fact, it's pretty rare that you find a membrane that's entirely a fluid nature. Generally, the domains of membranes are quite mosaic in their structure. So the fluid mosaic has moved more and more towards mosaicism as time goes on. And basically that's because most cells are organized in tissues, and tissues have very important asymmetric structures, and those are, at least on the membranes, are controlled inside and outside the cell by interactions. And even in 1976, we were aware of this, and this is a review that I wrote then, that there were various complexes and various domains. And these were actually regulated, controlled by extracellular matrix and intracellular cytoskeletal elements, uh, tubules, microfilaments, and other structures inside cells. And so these were important in terms of the static relationships of membranes controlling the mobility of components in the membrane. And so some of these elements can be quite complex. And this just shows Benny Geiger's idea of what some of these enzymatic complexes forming at the inner surface of the membrane into various types of complex structures. And these sort of complex structures are involved in a variety of different activities of cells. For example, motility, and this is shown here, that some of these domains are involved in attachment points to allow cells to attach, for example, the matrix into the cytoskeleton. And this allows cells to actually move on the basis of those attachments. So we know the biological membranes are asymmetric in the distribution of membrane components. And this is true with the basic lipid bilayer and the intercalated components into the membrane. But it's also true of the peripheral membrane components and membrane-associated components like the cytoskeletal components and extracellular matrix components as well. And I think I'll just skip this. This shows that the distribution, even on the inside and outer leaflet of the lipid bilayer, have differing compositions. And those are controlled basically because of their synthesis and enzymatic modifications, as well as some other characteristics as well. I'm going to just skip this. This has to do with the differences in formation of domains and specific structures and how they're important. And we know that the membranes are asymmetric, and I just threw this in because as a graduate student, this was one of my first projects, was to look at the asymmetry of membranes. And I had designed some ferritin antibodies that bound to spectrum and the inner surface of red blood cells, decorated only the inner surface, and using an influenza virus to showing only the outer surface. So we knew the membranes were quite asymmetric in their organization. And we now know that, of course, they're much more complex than the 1972 model. And in 1972, it looked like there was a fluid sea of lipids and everything was floating around, but that's not really true. In fact, they're much more dense. And as time, I've added this little arrow at the bottom here that they become much more dense, much more mosaic in their structures, much more static, particularly in certain regions. In most cells and tissues, for example, this is the way cells are organized. There's very little actual fluid in between these domains. So specific membrane lipids and proteins form these different domains, and this turns out to be a very common feature of most membranes in most cells. And actually the only types of cells that are oddball to this are the certain cells that are float in the circulation, for example, and other things. So these different structures can be organized in domains, and sometimes these domains are the reaction to binding of a ligand. And it just shows, for example, lipid rafts forming by a signal, the binding of a particular signal outside the cells, organizing lipids and proteins into complex domains, which constitute a signaling complex. And so most signaling complexes at the surface occur this way through the organization over time of more complex structures. So starting with more diffuse structures, even though some are basically immobilized, others are motile. And with time, some of these structures can reorganize to form different types of domains, which have differing compositions. So membrane components exhibit different lateral mobilities and distributions, as I've discussed. And these are functionally important because they interact in different ways. They form communication structures, for example, with other cells, with other ligands outside of cells, and so on and so forth. So most cells form different layers, like an epithelial cell layer, for example, which are by their nature very asymmetric, where in this case we have an apical surface, which is quite different from the basolateral surface in terms of the types of components that are there and their organization into different domains. And so, again, this is the more usual situation of cells and tissues, not that everything is all mixed up. There are also different lateral mobilities in cells, and this is kind of shown here from Ken Jacobson's study on labeling different types of reporters at the surface and showing that they have different types of motilities. Some are basically entirely random, others are partially random, that they'll start out being random, but they're actually confined to a certain region or corral, as they call them, where there's certain transmembrane proteins, for example, which form barriers. And then they can move from one barrier to another barrier. If one is randomly moving in the membrane, you don't see this type of movement of components. So we know that this occurs in the membrane. And if something is attached, for example, across the membrane to a cytoskeletal element, we could have this directional movement. That's usually an energy-driven process that requires cytoskeletal elements to contract, to move structures. And you see this, for example, in endocytosis, exocytosis, and a number of other very important properties of cell membranes. And so we have these different structures that are involved in not only communication, but in bringing components into cells, inside cells, or extruding them outside cells and releasing them into the environment. So cellular membranes are organized at multiple levels. We have the first level, which is the lipid bilayer of integral proteins and domains. And the second level are the peripheral proteins and various enzymes and so on, which are often attached to the integral membrane protein. Then we have cytoskeletal and extracellular matrix components, and these form the third level, or membrane-associated level. So we have this different hierarchical organization, and I showed this before, that these various domains, some may be mostly lipid with some protein, others may be mostly protein with some lipid into various complexes. And these form various barriers, as I mentioned, and this is shown here in a review by Kasumi. And I'll take a drink. Where these barriers are made up not only of proteins, but an annulus of lipids, and they interact with one another through various structures, and these can be cytoskeletal structures or other structures, but they form a complex matrix. And at the inner surface is shown down here in the lower aspect here. It seems to be very complex with interactions inside the cell. And so this hierarchical structure shown here sort of as a kind of a kabuki presentation, where the first structure is the basic bilayer and things that are intercalated into that. And as messages come in, for example, a second structure is then become generated, which is the complexes and the domains that form on the basic of that. And then there's actually a third level that forms within the cytoplasm of the cell. And so there are various ways of looking at this, but it's quite complex. So cellular membranes are really characterized by dynamic changes in structure and the formation of domains, and allowing them to efficiently react to environmental signals, and also to control the structure of membranes and structure of tissues in general. And basically, it's kind of a repeat of that, what I just mentioned. So the accepted basic model for membranes, which is really the bilayer basis with intercalated membrane proteins, is the Fluid-Mosaic model. Now, this has been updated a number of different times, and as time goes on, membranes have become more complex and more mosaic. That is, they have more complex components which are restricted in their distribution. But the general principles still apply when describing cellular membranes. And these domain organizations and lateral segregation components are extremely important in terms of the organization of tissues. And these complex dynamics allow cells to respond to extracellular and intracellular signals via transmembrane and lateral interactions and other complex situations. So basically, that's my presentation in a nutshell. And my kind of summary is the Fluid-Mosaic model is still alive, is still kicking, and is still useful, but it's much more complex than we originally proposed. But again, that original proposal was 52 plus years ago, and so naturally things are going to evolve.
Well, thank you very much. That was a great talk and covered a lot of ground and a lot of amazing history and contributions by so many people. I think those principles are really quite useful as well because they can organize our way through the complexity and some of the chaos that still might exist in our minds, at least in terms of membrane organization. I'm wondering, do you think it'll be possible to predict at some point how proteins get sorted to different membranes and assemble and become activated from first principles, from the birth of a protein on the ribosome to, let's say, functionalization on an endosome or plasma membrane or Golgi compartment? Are we getting close to?
I think we're getting closer, because basically the most important step, of course, is the synthesis of the membrane protein, because the way it's folded and way it's determines the way it interacts with lipids. And then the next step, of course, is the sorting of the various lipids to form an annulus around the membrane protein. And in the various types of membrane proteins, of course, determine where it's going to be in the membrane, whether it's an integral membrane protein, a peripheral membrane protein. And of course, I'm not counting the membrane associated because they're synthesized completely separately. And well, in fact, they're probably all synthesized separately, these various classes, but they do self-assemble based upon the interactions with themselves and with other components. And that first level of interaction, of course, is with the lipids of the membrane. And then the next level of interaction would be self-components like protein-protein interactions and so on. And of course, it's a very dynamic process, and that's important.
Great. Just to finish up, do you have any advice for students who are entering the field or thinking about entering, any gaps that need to be addressed or methods that you think should be applied to tackle the remaining problems in the field?
Well, I think there's still a lot of problems in turning how membranes actually signal and how those signals are actually processed. And all that's very important because the communication, cell-cell communication, is a very important topic. And it's pretty much a black box still. So that's one area of very important. Then the other thing is the hierarchical structure to build tissues. That's very important and is often overlooked. And the formation of tissues is, of course, an absolute necessity for complex organisms. And so those types of interactions and how we can still maintain that and still maintain the dynamic aspect of membranes when they're in the complex tissues is a very important problem, which has to be solved.
Okay, thank you so much. I'm going to stop the recording, but thank you again for a great talk and for answering a few questions. I really appreciate it.
You're welcome.